I'm going to begin my talk by asking a question. Where can life begin? Hydrothermal vents or hydrothermal fields? This is both by myself and my colleague Bruce Deamer. So you're going to hear something about both of our work in my talk. Most of the world's water is in the ocean, salty seawater. So it's been very obvious to people that life must have begun in the ocean, in salty seawater. I'm a biophysicist, and I have some problems with the self-assembly processes that must have occurred that accompany the origin of life. And salty seawater is really not the best place. So we are proposing an alternate, and that is freshwater hydrothermal fields. Now, let's take a look at the two kinds of water that we're going to be considering. First, we have seawater, and we have a source of energy that was discovered back in 1977 by Jack Corliss, and Martin Russell and Bill Martin and Nick Lane have been promoting this as a possible site for Earth's origin. But then we also have another kind of water, about 1% of the Earth's water today, which is freshwater hydrothermal fields associated with volcanic land masses. So salty seawater or freshwater in these hydrothermal fields. That's really an interesting question with a lot of leverage, because this also affects the way we think about the possible origin of life on other planets. So here we have an artist's rendition of what a hydrothermal field would look like on the Earth. There's a source of organic material, either raining down as meteoritic infall or as geochemical synthesis at the Earth's surface. This then accumulates in a freshwater hot spring, which undergoes evaporation and refilling on a regular basis. Those cycles of wet and dry are measured in anything from minutes to hours to days. Now, what about hydrothermal fields? Why would they possibly be a more plausible site for the origin of life? Well, for one thing, organic compounds would accumulate in hydrothermal ponds, as opposed to the vast ocean where any organic material would be diluted to micromolar concentrations. The hydrothermal fields today are generally acidic because of sulfur dioxide that gets dissolved in the water and produces an acid down in the pH 3 range. And also the concentration of ions are in the millimolar range, as opposed to the half molar range of sodium chloride in seawater. Wet-dry cycles are impossible, obviously, in bulk seawater, but wet-dry cycles on volcanic land masses can concentrate potential reactants. When they are concentrated, polymerization of monomers is promoted in the dry phase of a cycle. Elevated temperatures provide activation energy, and finally, if there are lipid-like molecules present, membranous compartments can assemble and encapsulate polymers to form protocells. So those are the components of a freshwater origin of life. Now, I need to just say something about a little bit of the chemistry that we're going to be talking about, and that is the chemistry of ester bonds. Ester bonds are synthesized by removing the elements of water from an organic acid and an alcohol. And an example that we all learn in elementary organic chemistry is that if you mix ethanol with acetic acid, it spontaneously forms an ester bond, and the resulting molecule is ethyl acetate, and water comes off as a byproduct of the reaction. What most people would not think of, however, is that the same reaction in principle can occur between the nucleotides of nucleic acids. Nucleotides are the monomers of DNA and RNA. And here I show one of these, adenosine monophosphate, where a phosphate group, which is in fact an acid, interacts with an alcohol group on the ribose to produce water and an ester bond. If you can make lots of ester bonds, you're going to make a very long chain of a nucleic acid. Finally, if we have a way to remove water from the reaction is drawn to the right, so you have an increased yield of the products. If we remove water from this reaction of nucleotide polymerization, again, you would, in principle, produce very long strands of a nucleic acid. So that leads me to a hypothesis, and that is that polymers resembling nucleic acids can be synthesized in freshwater hot springs. We've been testing that in the laboratory, and my talk is going to go through about 6 tests of this hypothesis. The first test is to expose mononucleotides to conditions simulating the wet-dry cycles we see in hydrothermal fields. The conditions in our simulation are a carbon dioxide atmosphere to maintain anaerobic conditions, elevated temperature, about 85 degrees C, which is in the range that we see in hydrothermal pools, an acidic pH range, and hydration-dehydration cycles. So we built a chamber. There's many ways to do this, but this is one of the ways that we've worked out. There's a little robotic chamber that has a rotating disc cycling reaction mixtures between heat and drying process, where carbon dioxide flows into little vials containing the reactants, and then that is cycled with a hydration by a little syringe pump that is pumping water into another part. So we have a choice of times. We can go anywhere from 30 minutes to a couple of hours, but a typical one that we're using is about 30-minute cycles between the wetting and drying. So here's a typical test. We're going to put some mononucleotides into these simulated hydrothermal conditions, and we're going to test whether the products can be labeled with radioactive phosphate. So we start with mononucleotides and a lipid matrix. If we want, we can add lipid and have membranes present. We do multiple wet-dry cycles, perhaps 4 would be a typical number. We get products, these are polymers that we can isolate. We label them with radioactive phosphate, and then we do gel electrophoresis. So here's the results. Over on the right, you see the little bit more detail about how we label them. Any RNA or DNA can be labeled this way. It's called in-labeling. You use an alkaline phosphatase and a T4 kinase plus radioactive ATP to add a radioactive phosphate to the end of an RNA. We did this with our products, and we were astonished to discover that, in fact, we had made polymers that were recognized by these 2 enzymes, were labeled with radioactive phosphate. And all of those dark streaks that you see are in a gel called gel electrophoresis. And you also want to take a look in the A up above, and you see what is called a ladder. The ladder is produced by known lengths of RNA or DNA. So we start with 10 mers and go all the way up to 100 mers of this ladder. And that gives you an idea of the range of polymers that we're seeing. And you can see that as we do more cycles, we get more and more polymers. All the others, I won't take the time to explain all of that, but in fact, this is a very robust reaction, and we've seen this time and again. So that's the first test. Here's the second test. We expect those polymers also to be labeled by fluorescent dyes, like other nucleic acids are. Here's a gel in which we've used ethidium bromide, an intercalating dye, and off to the left in part A, you see 2 known polymers of RNA. One of these is called poly-U, polyuridylic acid. The other is poly-A, polyadenylate acid. And you notice that they don't really label with this dye until you mix them together. It's well known that these 2 will produce a double helix. The ethidium then can intercalate between the bases of the double helix and is strongly labeled. And then clear at the right, you see a streak labeled product, and that's in the same range as the radioactive products I showed you earlier. Here's another dye clear to the right called CyberSafe, and you can again see at labeling a product. Test 3, if the polymers are nucleic acids, we should be able to sequence them using nanopores. We have nanopore sequencers in the lab, and this is how they work. We have a double strand of something like DNA, for example. It's a double-stranded helix. There is an enzyme called a helicase that unzips the double strand and allows the single strand of the DNA to advance through the nanopore at about 450 nucleotides per second. The nanopore in this case is called CSGG. It's a bacterial porin, and that's embedded in a lipid membrane. As it's pulled through by an applied voltage, we can see picoamp modulations which are specific for the bases in the limiting aperture of that pore. That then allows us to sequence any nucleic acid that we want to, both DNA and RNA. I'm going to show you DNA because it's easiest to do with DNA. So here's how you do it. You start with thymidine monophosphate, which is one of the monomers of DNA. We put it through multiple wet-dry cycles. What we expect is oligothymidyllic acid, a lot of thymidines in a row. We then analyze this by ligating it to a known DNA. These oligomers are too short to work by themselves in the nanopore, so we ligate it to a longer known DNA and then do nanopore sequencing. Here's some DNA being added to a minion. This is exactly what we do. The minion is attached to a laptop, and the laptop has a software that analyzes the electrical signals that come off and calls the bases. So here's a typical signal from a single molecule going through a single channel in the minion nanopore sequencing device. Off to the left, you see a lot of up and down traces. Those are caused by the advancing nucleic acid through the pore. Then you see a point where it goes down and it's labeled oligo-DT homopolymer. That is where we would expect to see an oligo-T going through. When we do a gel of that oligo-T, we can see individual radioactively labeled bands clear on the left. That's the gel. And then the white trace against black is a scan of that gel. And you can see that our labeled material goes all the way from below, around 30 mers clear up to a little over 90 mers. That's the same length that we see in our gels. When we do base calling on the using the minion, we discover that there are long strands of oligo-T attached to that strand of DNA that is carrying it through. The shorter strands are down in the 30 mer range with that arrow pointing to it. Medium-sized strands are in the 40 to 50 mer range, and the longer strands are up around 90 mers in length. So we can conclude that we did in fact synthesize a molecule that resembles DNA in all of the tests that we've done. And particularly, it is recognized as DNA by the enzymes used to analyze it. Test 4, we expect the polymers to be encapsulated if a lipid is present. On the left, we see RNA after it's been synthesized in the presence of a lipid. And then in B, you see a fluorescent stain showing the brightly labeled product in the vesicles in that same image. We have a phase image on top and the fluorescent image down below. Off to the right, we've made DNA with the lipid present and labeled it with a DNA stain called DAPI, D-A-P-I. And you can see the strongly stained vesicles containing what very much looks like DNA encapsulated in the vesicles. Test 5, we want to try this out in the wild. If it works in the laboratory, it should work in the wild. So we have made 2 trips to Hell's Gate, and we have run the same wet-dry experiment using the water from this hot spring and the temperature range in this hot spring in Hell's Gate. Here's the vials that held the reactants. You can see that. That was immersed down in a hot area, a shallow area, of course. We didn't want the hot spring water to get into the bottles. And then we brought that home and analyzed for for RNA. And we found that RNA-like polymers were synthesized. Those look exactly like what we see if we did that in the laboratory. Here's a gel. On the left is a laboratory version showing the product, and on the right is the products from the hot spring condition. Finally, we wanted to visualize the polymers, test 6. We were surprised to see ring-like structures mixed in with lots of little particles, which we assume are the shorter strands of RNA in this case. This was just put up on bioRxiv, and you can read the paper in bioRxiv, the April edition, if you want to. My colleague there was Sue Hassenkam. He's at the University of Copenhagen in Denmark, and did the AFM. So all of this comes down to a summary, an alternative scenario for life's origin. Life begins in freshwater hydrothermal pools subjected to wet-dry cycles. Life did not invent nucleic acids. Instead, the chemical energy made available by wet-dry cycling generates random RNA-like polymers. If amphiphiles are present, the polymers become encapsulated and produce vast numbers of membranous compartments we call protocells. Each protocell is different from all the rest and is in essence a microscopic experiment. Populations of protocells can undergo selection and evolution. Now, the last statement is basically conjecture. This is for the future. If we are right, this proposed alternative to be tested is that life began when selected systems of encapsulated polymers emerged that were capable of catalyzed metabolism, growth, and replication.